Okay. And I want to sort of say that this started by a telephone call. This often happens. And the way this went is that Susan was leading clinical pharmacology at the Princess Alexandra Hospital. She was the senior person in medicine, great passion for liver, great passion for therapeutics, and a great passion for toxicology. And the reason she approached me was I'd done some work trying to change the thinking about how we manage liver disease using chemical engineering principles. And Princess Alexandra Hospital was the first site in Australia to do liver transplants. So she thought I'd be a good addition to their mix. And at the time, I was a disillusioned young professor, head of department, effectively thought I'd retired at 38. There was a chance to come back home to my family. And when I asked my wife, she asked, can we go tomorrow? And this was giving up a tenure job to become a research professor with an uncertain future. Every 5 years, you have to reapply for a job or get money. It's a tough gig. But this was sort of a wonderful opportunity. And Lions played a role initially, which I had to apply for, in getting me started. And I had support from some other friends. So I had initial appointments at Bond University with Peter Andrews and in pharmacy with Peter Stewart. So what I'd like to highlight from the outset is we've been a very successful group. So I just chose my first three graduates. One is the Professor of Neuroscience and Head of School at Griffith. A second is the Pro Vice Chancellor of Research at University of Sydney. And the third is Professor of Geriatric Medicine at the University of Sydney. And some of my students are Deputy Vice Chancellors of Research and a whole range of different positions, and we're very proud with the outcome. And in fact, Mark Smithers and I had dinner the other night with Zhenya Wu, who showered us dinner. It's not often that you have your students looking after you in style, and he looked after us in style. The other part, though, is that because Sue was interested in the liver, we got interested with working with the hepatologists at particularly in rural Brisbane, Laurie Powell and Jean Halliday. And one of their PhD students was a young Darrell Crawford. And in fact, I'm able to show you, this is actually an extract from his PhD thesis. And in fact, this led to, and I still remember doing a mock interview for him for NHMRC, which I think he got. I put him through the ringer. He didn't know what he was going through. But we've ended up with a long, long association as a result of all of this. And so the key thing which I introduced to this field was to use chemical reaction engineering. It's interesting because I'm using the similar principles now with the FDA, but with skin, but in liver, to use chemical reaction engineering principles and try and understand what happened to the liver by doing what's called impulse response relationships. And at the time, everyone used steady state infusions, but if you use this technique, you can actually sort of, with a perfused liver, you can actually collect data that sort of tells you the time course of how the drug behaves after you distribute it. And what this has done is allowed us to separate out how drugs cross the hepatocyte wall, how they go into the bile, how they go into the individual organelles, how there's differential transport into, say, mitochondria, into other cells, such as lysosomes, and also how they're affected by things like fibrosis. And one of the things that Darrell's group did at this time was provide to us a whole host of liver diseases and advise us upon what liver diseases do we need to study that we didn't have information for. So this was really very valuable. And in fact, I think one of the things that Darrell may mention, if you want to ask him questions about, is he had the special MDR2 minus mouse, which had a very special sort of progression of liver disease. And we used this and we could actually sort of predict from the physicochemical properties and from sort of a histological measure, which we use computerized imaging to calculate, called fibrosis index. We could actually work out what the likely uptake would be of drugs into the liver, which is really an issue in things like liver fibrosis that occurs after either cirrhosis or can be after viral disease, particularly hepatitis, or can also occur after non-alcoholic steatosis. And we could also look at the same time what role did transporters play in this and how were they affected. One of the key outcomes from this is that there's a whole heap of people who you may recognise on this list who this work fed into. because this work, having started perfused livers, we could start looking at perfused other organs. And so you'll see some work I'll show you in a minute on the skin. I did some work with Mark Smithers in humans on melanoma, pancreas, with Paul Colditz in terms of the brain and neonatology. A lot of work with Peter Kruger, Jeff Lippman, Jason, my son, and Bala Venkatesh, and some work with Laurie Hurst in ophthalmology. And you can see from this, the last paper, that we're still very productive in this space. And one of the things I learned in this early on when I first visited to Linköping, where I met Chris Anderson, who's an expat Australian, who's a professor of dermatology there, was he had these catheters in my arm within 1 hour of my arrival. So clearly I was a volunteer and clearly I was into this. This was a very important breakthrough for us in Queensland. So our work was published in The Lancet. What it showed was when you apply things like Dankarab or Voltaren gel, compounds do go directly into deeper tissues because they're shunted deeper tissues by the blood supply. And our work showed that. But what we could also do is use this to look at, can we get selective drug delivery into melanoma? So we could use this to try and measure the effectiveness of isolated lymphofusions to treat melanoma without doing these perfusions, which John Thompson in Sydney and Mark was doing here, and Bill Edgerton really sort of started off this technique. You would have almost no chance of survival in those days. I think we've got some better drugs, but this is still a common technique. And the other place where it's used a lot now is in critical care medicine, because a lot of infections actually arise from the skin. Oftentimes in sepsis, that whole area becomes poorly perfused and you have poor uptake. And so you need to know whether your levels in the skin are high enough to give you sufficient antibiotic cover so that you actually can recover from the sepsis. And we've had a number of papers in this area on burns, on sepsis, infections, which involve those people I spoke of earlier. And this work has been carried forward. We have a current NHMRC grant, which Tina Liang and Halu and Darrell basically helped driving, along with Gordon Zhu, who's over at over at AIBN, where we actually have also applied pharmacokinetics. I'm not going into the details. I'm just going to give you an overview. But basically, by using these techniques and by using imaging, we can look at where things go and we can describe them in really quite clear terms. But I want to highlight one important aspect, that when I look back at all the breakthroughs we've had, the breakthroughs that had the greatest impact did not come from the NHMRC grants I had. They actually came from more like MRFF-like grants. And I really would encourage people after the NHMRC results, where so many people are depressed, to get into the MRFF scheme. There are so many grants out there that you can go for, and you can make such a difference. Please think about it. So a lot of this work we did was randomised controlled trials. It involved a number of these people used to be at UQ, people like Chris Delmar, Andrew Rawson, Paul Glasio. Sean, unfortunately, has passed away. And there's lots of other people who played a key role in this. And this work has led to the federal government funding this nationally. It's rolled out as residential care. It's got guidelines. It's in the home. It's in everywhere. Dose administration aids. You'll probably see it every time you're walking around. These are some of the impacts from that work. There's now a review, as you know, of the nursing home sector, and clearly I think some of this has to be revisited. So oftentimes people lose their sort of, I can call it the momentum when you don't review things regularly. And I think probably this needs to be sort of developed. So there's opportunities in this space, and I encourage everybody to be thinking about it. There's other areas that we got involved with. Ian Fraser and I worked with Mark Kendall in the area of Vax's nanopatch. We did a lot of work together. One of the key things we showed is that buccal and skin was probably more effective than intramuscular administration for some of these agents, and clearly that oral wasn't going to be effective at all. I'd also want to highlight, and probably I should highlight Colin Page. He's actually passed away now, but he was director of the clinical toxicology unit at PAH. We did a lot of work with that group, basically trying to help them deal with poisonings. This is one example of a poisoning that we advised them on, and they used basically dialysis, and we measure both the levels and the response, and you can see the two parallel each other. And the sort of the outcomes of this is really sometimes unexpected. So one example of sort of an outcome is this work that was done with Nick Buckley, and this has just been published. And what this has led to, if you look at the figure carefully, and I'm not sure this can be seen, maybe I'll try it with this. You'll see, if you look at this carefully, No, it doesn't show up. There's a gradual drop off in the actual level of poisonings. And what's happened is that our work has led to the banning of various pesticides worldwide. And when those pesticides have been banned, then the actual deaths fell away. So what our work was trying to do was identify which pesticides are most likely to kill people, because oftentimes animal studies do not translate into people, and then get those particular products banned. We've done that, and this work published with Nick has actually been sort of well taken up. I also want to sort of say that one of my real passions I've developed in this area is actually for imaging. And TRI is a great place for imaging, and we've done a lot of our imaging here. And a lot of this work, for me, one of the satisfactions is that my students have also adopted this. So I'm going to give you two examples. One by Yusuf Mohammed that he led on sunscreens, on zinc oxide sunscreens, which has actually led to the FDA now having only two generally regarded as safe sunscreens available. One of them is zinc oxide, which our work contributed to because we showed that on repeated administration, this compound is not absorbed into the skin. And you may be aware there's lots and lots of hype out there from people trying to create grant money by saying nano products are dangerous, when in fact some are and some are not. What really matters is their intrinsic toxicity, number one, and secondly, are they actually absorbed? Because if you're not exposed to it, it's not going to cause you problems. And when we actually when we could use cell behaviour to actually show that these were in fact, so this is things called fluorescence lifetime imaging, and you can use changes in redox state to show there's actually no toxicity. The second area which I'm going to highlight is from Tina Zhang, who's actually in the audience. And this is some beautiful work she did looking at the uptake of nanoparticles in the kidney. And what she showed was nanoparticles of a certain time get trapped in the glomerular mesangial cells. So this is important because what it says is certain nanoparticles should not be given to humans if they're not biodegradable, because they can be trapped in the kidney if that's where that's excreted to. And the particular size that we need to worry about are particularly those which are between around about 50 to around about 100 nanometers. The other thing which we're now doing is we've used a lot of this imaging to go another step further, and this is work that's been done with Tina, and Tina's now leading all this work. I've stepped back. Darrell's also stepped back, but you'll see a lot of her PhD students are in the audience, and they've actually participated in this, and also involves people in Sydney. But we've actually used this sort of imaging now to start looking at trying to characterise the microcirculation, but particularly related to my poisoning work, what happens when you poison things. So this particular case, if you take high doses of paracetamol, it will actually cause liver toxicity. Most of that occurs in the actual central vein, around the central region. This is the inside part of the liver. And what we're trying to do is understand how that occurs and what that time course was. The other thing we've done while being at TRI, I'm not going to go through the details on the slide, but the guys at QUT have been fantastic. We've worked closely with them in trying to work our stem cell work up, and they've helped us with great generosity in actually trying to relate this to what we're doing. And we've got lots of work going on with them, both with their mathematicians and with their sort of cell biologists. Fantastic, really happy guys. Lastly, we're working with AIBN in a range of areas, and a lot of that work deals with what I call lab on the chip. So over there, we have two of our PhD students working collaboratively with AIBN, and what we've been doing there is trying to look at, can we create basically an artificial model, so rather than use isolated perfused livers, rat livers, actually make human livers, but actually on a chip, so we can study them directly. I'm now going to move on to Darrell. Darrell?
Thanks, Mike, and thanks for the ride. As Mike said, we've been working together collaboratively now for decades, really, and there's been sort of so much help to a number of people that I've worked closely with, and it's been a real pleasure. So why liver disease, and why was I able to get Mike a little bit interested in all of this? Well, because this map shows the the sort of global prevalence of liver disease. Cirrhosis, 11th leading cause of global deaths, about 1.32 million deaths per annum. And you can see that the higher prevalence occurs in some heavily populated countries. And so that is really what contributes to, by and large, to this extremely high level of liver disease in the world. So, for instance, there's 271 million people with chronic hepatitis B, and there's 71 million people with hepatitis C in the world. It's quite interesting when you look at trends in mortality over time, and in fact, globally, you can start to see a decrease in liver-related mortality, and that probably relates to public health measures, vaccination, better understanding of transmission of hepatitis B. But in other parts of the world, in other less well-developed parts of the world, the mortality remains increasing. And we see here that the causes of cirrhosis-related deaths across the world in men and women, and see hepatitis B, almost a third of cirrhosis deaths across the world are related to hepatitis B, a quarter to hepatitis C, alcoholic liver disease, about 27% in men. And we've seen over really the last 30 years the emergence of fatty liver disease as a real entity, now accounting for, when you look at the two-pot, between 7.7 and 11.3% of deaths. So, 40 years ago, you could write a paper on fatty liver disease as a case report almost, but now it's so common. And then other liver diseases, including iron overload, which we're interested in, between 8 and 17%. So, and to continue the fatty liver disease story, we're now talking about 1 billion people in the world having what we call metabolic associated fatty liver disease, which is a recent change in terminology. And you can see that the high prevalence areas are directly related to the calorie intake, the average daily calorie intake of individuals. And so, in the States, where you're looking at about a quarter, South America a third, as socioeconomic conditions improve in parts of Asia, you start to see this big increases. So we've really seen this disease emerge over the time that I've been in practice. And to continue Mike's theme about therapeutics, what we are interested in is that we often see patients, so our interest is in this disease called hemochromatosis, which is an iron storage disease due to increased iron absorption. And this disease is due to a deficiency in a small peptide that is the master controller of iron balance called hepcidin. And this paper is from Kim Bridle, who's obscured by me up here, but Kim published this in The Lancet. It's almost got 500 citations. I checked yesterday, this paper. And so, but we see a lot of people who've got iron overload who have got obesity. And the interesting thing about obesity, and where we've got some researchers at the present time, is that this visceral fat is not an inert, inactive sort of tissue. It's actually very metabolically and active. It's very pro-inflammatory. And this makes a number of pro-inflammatory molecules that, because of its location, it drains straight into the portal vein, straight into the liver. And so this sets up or contributes to the inflammatory liver disease known as NASH or MAFLD. Interestingly, in our animal models of this condition, you also see a reduction in hepcidin synthesis or expression. And one often sees derangements of iron metabolism in this disease as well. And then collectively, these conditions, we think about 30% of the patients we see will have what we call co-toxic liver disease. And these amplify each other's response and put patients at higher risk of cirrhosis and development of liver cancer. So we're very interested in looking at hepcidin therapeutics to try to restore this balance here. And it's interesting because when you're deficient in hepcidin, you export iron out of cells. So particularly the small intestine, you have rapid iron export, and that's why you get iron overload. But iron is also exported from other cells, including adipocytes. And so we're interested to see whether the administration of hepcidin inadvertently causes an increase in iron content of the adipocytes and what effect that has on pro-inflammatory mediators. So this is really just looking at a few IL-6, which is obviously a pro-inflammatory molecule, adiponectin, which is a protective molecule, and this is the addition of iron just in cells, and you can see you get an upregulation of IL-6 and downregulation of protective proteins, and this is mirrored again in those when you treat these cells with hepcidin. And so we're continuing this work. We've got parallel animal experiments looking at hepcidin in animals with co-toxic liver injury, and it's just going to be really interesting, I think, to see whether we inadvertently actually potentiate injury by activating adipocytes through the inadvertent increase in cellular iron concentration. Now, where there's liver cirrhosis, there's liver cancer. So this is really truly an emerging epidemic in the world. This is taken from the publication in the European Liver Journal. And you can see where the increases across the world are pretty dramatic. The Philippines, for instance, over about, I think, a 5-year period, 7-year period, or maybe a 10-year period, up nearly 900%, a lot of that due to better diagnosis. But Australia, even a really dramatic increase in the prevalence of primary liver cancer. And you can see sort of globally, you see the change in the cancer cases really all across the world. And when you look at worldwide estimates of liver cancer, one of the interesting things is you see how common liver cancer is as a cause of mortality. And interestingly to me is this almost one-to-one relationship between the number of deaths and the number of incident cases, really telling us that our treatments are very, very poor and that we make the diagnosis late. We've got very few therapeutic avenues to utilise. And then when you look at local data, so this is the Australian data, where you can see this dramatic increase in mortality from liver cancer occurring over the last 2 decades, both in men and women. And this is due to the emergence of hepatitis C, it's due to the emergence of fatty liver disease, it's due to migration patterns of patients with high prevalence coming from areas of high prevalence of hepatitis B, it's due to continued alcohol consumption. So this is increasing, this tumour is increasing at a faster rate than any other cancer in Australia at the moment. And interestingly, which is sort of, at some stage you have to drill these things down to your local population. And this is really a paper we had around looking at remoteness, race and social disadvantage in regard to HCC incidents. And I draw your attention down here that talks about the poor survival being associated with greater area level social disadvantage, older age and less recent period of diagnosis. So if you live in remote areas, regional areas, particularly regional areas with a high Indigenous population, your overall survival is significantly worse than those people who live in metropolitan, Caucasian-dominated populations. And this is relevant because some of our work is around diagnostics, but this emergence of liver cancer as an epidemic and our great collaboration with Mike and his group really led us to forming a partnership between Glipley Research Foundation at Greenslopes and Diamantina, really around liver cancer, and Tina and Harlu have been instrumental in that. And basically we cover a spectrum of research programs. We have got studies on the sort of pathobiology of liver cancer, diagnostics, therapeutics, and issues around public health. And so one of the projects that's very interesting around pathobiology is Tina's project, which is about tumour self-seeding cells. So Tina can transfect cancer cells with a KIK-GR plasmid, which when you expose it to ultraviolet light turns red. And so she's able to see cells, cancer cells, then inject that into livers and then look for migration. And then you can see this population of cells rehome back to the primary cancer in the liver. And so Tina's been characterising these cells, looking at having developed this novel self-seeding tumour model with transcriptional characterisation and functional characterisation of these tumour self-seeding cells, which is a really interesting concept and novel in liver cancer biology. And so in addition to that, we've got a lot of studies going on the microenvironment for liver cancer. Our diagnostics, we've got a really interesting project based working with Jamindi Pandaria, who was based with QUT here, around salivary diagnosis for diagnostics for liver fibrosis and HCC, which one of the problems that these regional populations have in liver diagnosis, because they don't have any access to diagnostic services, and the liver biopsy, non-invasive tests of liver fibrosis just not available to people once you get about 150 miles out of Brisbane, and so their liver disease and their liver cancer was diagnosed late, and so saliva-based diagnostics really provides an avenue for those people to have their diagnosis made earlier. We're doing some work with John Hooper of CDCP1 as a diagnostic treatment marker for bile duct cancer. Tina's work on therapeutics and G's work on CD14 targeting for underlying liver disease. And then in public health, we're part of an MRFF that we just recently got on risk stratification pathways for surveillance in primary care. And then this whole issue around saliva, improving access to diagnosis in disadvantaged communities. So I want to stop there and just let Mike close this session.
Yeah, so I thought I'd finish off by sort of asking the question, so what have we got to show for all of this? What have we got? So I think that sort of we've got a lot of PhD scholars. We're very proud of them all, and you can see some of them in action here today. We have a number of publications, and we have been awarded 5 NHMRC project grants to date, which I think is pretty good. But I think for me, even more exciting is to see what's happened with our graduates. And we take great pride in those. One of the sort of sayings I have in life is don't judge your performance by how you do, but by how those around you turn out. And I'm very proud that, and Darrell, I think both of us together, that if we look sort of at our collaborators and how they've turned out, that we have something like about 4 or 5 actually what NHMRC investigated grants in the last round. This round has passed. And so they were Nick Buckley, who does all the clinical toxicology with me, Bala Venkatesh, who does a lot of the intensive care work at the Wesley, Jason, my son, Chungsia, who's at AIBN. She's now going across to Adelaide because Peter Hoy convinced her to go over there. So it's a loss, but she and I are working collaboratively together. And because I'm in Adelaide as well, then that's really a wonderful collaboration. Claire Roberts has works on placenta. We didn't talk about placenta today, but I've done a lot of work in that space. And Yusuf won an FDA sensorial grant. And of course, DI has been very kind to give Jeff Grice an honorary position. So I'm very lucky, and we both are, that people have done so well. I want to highlight, and those which are circled really are 3 of the key players in the liver area. Shin Lu, who's had a fellowship tour quite recently, and who has been really a mainstay for me. When she first came here, I still remember her first arriving, could hardly speak any English, but has been a wonderful, wonderful member of my team. Tina and Halu met in the lab, became husband and wife, had children here, convinced Halu he should become a clinician scientist. He's now doing his medicine to do that. He's a liver surgeon by training, and I think, Darrell, that's a wonderful part for our future. And of course, I highlight again that we have others. But there's also others. Maybe I'll let you talk about the last slide of the group, Les, and all the people out there. Maybe you should finish up with this comment on notes.
So this is our group. As I spoke about, Kim Bridle, who has been with us for a long time and really driven a lot of the laboratory work in this, and Halu and Tina that Mike's spoken about. We've got scholars really from all over the world, which is fantastic. We've got probably, between Mike and ourselves, I think we've got probably the most multicultural research group. I think I'm the only Australian. And Kim, really. But it's great to have people from all over the world. Falabi here is from Nigeria, and he's working with Kin on the Hepsidin project. So I'd just like to close there, Mike.
Can I make one last comment?
Sure.
So one last comment I want to make is, you can see the picture up on your left-hand side. This is my group on a retreat. And I'd really encourage TRI and others to do this. I used to use my, industry funding. I'd take people away to various resorts such as Noosa, down to Byron Bay, across to Stradbroke Island. You probably remember some of the Shin and Tina. And this was about trying to build culture. And they'd take their families over, and we used to organise a day that they would work in the morning and at night, and during the afternoon was completely free, have a barbecue, and we tried to build culture of interactions. I think this is really important. I don't see enough of it. One of the great things, Darrell, I remember when we first had was what I call the esprit de corps across medicine. We had all of us meet from PA, from Greenslopes, from Royal Brisbane, from Prince Charles. And a lot of the collaborations I talked about, Dave, came from us just meeting after staff meetings, having a cup of coffee and getting to know each other. I think there's an issue we have right now about being isolated, and I think there's opportunities for us in the future. But thank you all for listening.
Thanks, Mike.
Yeah, Darrell's going to run that. I'll step back.
Okay, some questions.
Yeah, no, he's on top of all this.
So I don't know whether we've got any questions yet.
Does anyone have any questions from here? Yes.
Hi. This might be a bit vague. It sounds like you've done a lot of stuff in both humans and animal models. Have you found any particular challenges in translating between those models? You did comment something about things not always working the same in people as in animals. But do you have any examples of things that?
Yeah. So, for instance, if you take the liver, a lot of the actual metabolizing enzymes in a rat liver is very different to a human liver. I'm sorry. In fact, Darrell knows more about this than I do. Do you want to comment? Yeah, so a lot of the enzymes, metabolizing enzymes that are in a rat liver, they're not the same as in human liver. And so what we're doing more and more now is, and a lot of my work that I was talking about earlier that they brought me here for was take hold of human data, human liver data, and this is basically isolated cell data, and then actually put it into a virtual human. So we can actually predict what actually occurs in a virtual human for a new drug by understanding how that drug is handled by cells. If you take a rat and do that, you can get very false information. Rats often metabolize things much faster than humans. And you can actually show that metabolism can vary depending upon the size of animals or whatever. And so you can't always translate animals to humans directly.
Yeah, then you don't have the same scenario where you can model the impact of injury or fibrosis unless you can. So you're talking about isolated hepatocytes or?
So what we're moving to is actually using isolated human hepatocytes on an organ and chip, and then incorporating that so we can actually construct the whole human body, if you like an engineer would. So we can actually put in what the blood flows are, different organs, what the actual volumes of those organs are, how it binds in those organs, the permeability, and we can do a lot of that by in silico modelling. What we have to do is we have to populate that with data, and then we have to validate it. So what we're finding more and more, and a lot of this is driven by things like the 3 R's, sort of the people trying to not do animal experimentation. There's also, and what we're trying to do is use a sort of almost a hybrid between cells and computers and human data.
So can you build like a liver dialysis scenario?
So the problem with liver dialysis is it causes livers to bleed. Do you want to talk about liver dialysis?
No, I was going to see if Tina wanted to comment on the chip and see whether that answers any.
Yes, I think currently we have the liver on chip. MSC derived the functional liver cells. So we have the 3 compartments with liver hepatic cirrhosis compartments and with
Tina, just talk here because people online can't hear you. Just that again.
So I think currently we have the MSC derived liver on a chip. So all the functional cells, the hepatocytes and the endothelial cells, as well as biliary cells, were all derived from mesenchymal stem cells. So we have the 3 compartments from sinusoids to hepatocytes, as well as secreted into the bile duct. So it's like if we have the patient, so we have the fibroblast. From the patients, we can differentiate into a real functional liver on a chip. So, yeah.
Yeah, and I should add, one of my real loves is actually mathematical modelling of that sort of information. So I'm one of those few people who tend to be interested in the biology, but also interested in the modelling. Any other questions? Oh, sorry, at the back. Yeah.
I'm Michael and Darrell. That's a very nice. So I come late, but I was listening the Zoom meeting. I have more, it's about collaboration and the translation. You have, I mean, you have shown great cases of lots of collaboration with the different disciplines. So I have a question around when we have this collaboration, sometimes it's because we have a question, right? We actively seek. People have a different skills and complementary activities to solve the problem. Another type of collaboration would be just have a coffee, we have a little brainstorm, we think together, okay, something can do together. So in your long-term experience, which way you really have a better outcome, and what's your advice for seeking collaboration?
Can I give you an honest answer? so in essence, you've got to find those people who you bond with. And you find those people you bond with, you can really do things. I mean, Darrell and I work like that. We don't expect anything of each other, and we just get great thrill out of working together. There's some people who will offer you the world but do nothing. So you have to really pick those people, and they'll pick you because together you're actually better than either of you by yourselves. And I think that's the essence. Do you want to comment?
Yeah, well, I think so too. I think probably if it is if it's an either-or question, which one's better, I don't think that you're going to have as effective collaboration as what you can have unless you have the coffee. I think that, I think establishing the relationship and I think probably, in business, I think that this is what sort of, the best businesses are ones where there's a fair bit of chat over coffee rather than being very, almost too strategic, if you know what I mean. So I think that sort of fellowship, friendship factor around research is really drives great collaboration.
Yeah, I think it's actually helping people in more than just the research. There's other things that you and I help each other with all the time, and it's actually in our lives. So I think it's about, it's about bonding.
Yeah, my own experience, I totally agree with that. More productive with people have a trust that we can, discuss all the time. And in terms of translation, sometimes we have a very. That's my question actually comes from. We can have a question. We need some kind of activities in our inner cycle. We don't have those kind of close relationships. And that's actually challenging, right? So you have to see someone we don't know each other, and particularly if we have clear, focus or aim or problem to solve. Let's say how, because this series is about translation. So when we have this translation, we have towards more defined aim, but a lot of this friendship oriented collaboration, this kind of come from, okay, what we can do. It's not what we have to do.
Yeah, I think
so that's the question about.
I think the other issue is, I mean, my observation is Paul's done a fantastic job here in TRI in terms of trying to build a culture here and trying to build that up. But what I said to you is, what really was been the great strength for me for collaboration is the interaction with all the other schools, with the other professors. The spree, what we had was this wonderful esprit de corps, that we could come to each other, talk to each other. We don't have that same esprit de corps right now. It's one of the things I really miss across the whole city. And so you'll find there's people in our, some of the people in DI aren't really my area. But the people, for instance, in QCCR, in intensive care and Wesleyan intensive care, when there's Peter over in, the hospital here, they are. And each of them will bring different things to you and you to them that others who might be in the same area can't. I mean, you may want to comment as well.
Yeah, I think the translation issue is built around context, I think. You do have to realise that there's a boundary to what, to what most people can do, whether, in terms of translation. You can only take things so far until you need somebody else to build upon, and often that person needs somebody else to build upon, to really take, say, discovery right through, well, to say, to market, that, the basic scientist, you might do this and the clinician might build on that, but then you need people with other skills. And finding that cluster of people that you trust and that you want to work with, and there's a sincerity in the effort, sometimes that can be challenging, can be difficult,
The other part of that is, do you know about the management wheel? So the management wheel says that each of us have got different talents, main talent and secondary talent. But you oftentimes will find someone who's a good explorer, promoter, innovator isn't oftentimes a good dot of I's and cross of T's. And you have to really try and realise that really some of the best people to be collaborators with you are actually opposite to your personality. And they can fix things up that you can't do. And I think it's one of the biggest things I learned in life is don't always try and find people like yourselves, but rather people who are different to you. If you can meld with them, they can really add something to it.
Yeah, I mean, there's a whole literature now about bias and how in interview panels people tend to select the person for interview who's just like themselves, which of course does no good for the business because then everybody ends up the same.
Yeah.
So you need the diversity. Is anything more on the chat? No. So
we thank all of you for coming. This has really been sort of quite special for us. Darrell, do you want to have the final word?
Well, just the final word, Mike, about your contributions over an extended period of time. There's so many people that you've worked with and that we've enjoyed working with you. It's so funny with Mike, you have to submit an NHMRC grant at 12 o'clock at night and 11:30 at night to get the final corrections. The grant totally rewritten.
Yes, I've had the special honour with UQ that I always used to put grants in late. Paul, you want to hear this? And what they said was, so what they used to say to me was, look, your hit rate's pretty good. We're happy to accept you coming in late. And I've had this experience. There's one particular trip I remember being on a plane. I was actually more on the plane than I was at the conference. Arrived at this conference and someone said a couple of things and I came up with this brilliant idea. Wrote a grant within 1 week and got it. And I thought to myself, basically, you've got to be careful with these grants. They become so manufactured and so, attention to detail. You lose the pizzazz. And that really sometimes can actually win you the grant. So we've got to be very careful in trying to become, just too controlled. Anyway, that's my last parting word.
All right. Well, thanks everybody for coming.
Yeah, thank you all.
Grab a picture of you two.